Cargo encapsulation by nanoshells has several applications, from nanomaterials to medicine. Recently, researchers at Brandeis MRSEC and collaborators introduced self-assembling DNA origami capsids as great candidates for these applications. But to achieve robust encapsulation with high yields, a thorough understanding of shell assembly is required. It's also important to know how the properties of a cargo affect the assembly of its encapsulating container. At Hagan Lab, we learn these assembly mechanisms from the nature by studying viruses and bacterial microcompartments. We develop computational and theoretical modeling to understand the inherent properties of these natural cage-like architectures and identify the key factors that control the assembly pathway and the efficiency of cargo loading. But will the cargo encapsulation change the shell size? Our dynamical simulations of shell assembly in the presence and absence of cargo showed that the presence of a cargo can either increase or decrease the size of a shell relative to its intrinsic spontaneous curvature, as seen in recent experiments. These results and our collaborative research on nanoshells will be a guide for designing efficient drug delivery vesicles and nanoreactors.